Welcome everybody to this parallel track number 7, which is a mixture of presentations for papers submitted to data and genomes track. So with no further ado, we will start with the first speaker, Qiao Liu from Tsinghua University. Qiao, whenever you want.
Yeah, thanks. Okay, thanks for the introduction. Hello guys, I'm Qiao from Tsinghua University. Today I would like to introduce one of our recent works for the cancer drug response prediction with a hybrid graph convolutional neural networks. We proposed a deep learning model to solve the cancer drug response problem. And okay, let me control. Oh, I have some problem control the screen. Sorry. Oh, okay. I'll mainly talk about the following 4 parts. First, I would like to talk some background information about the cancer drug response. And second, I would like to illustrate how we build our model, how we model the biological problem. And the third, I would like to introduce some major results achieved with our deep CDR model. And last, I would like to make a very brief conclusions about this talk, and hopefully there's still some time left for discussion and questions. Okay, so let me first talk about the cancer drug response. The cancer drug response actually is a problem which describes how a cancer patient can respond to a specific drug. You can use several words to describe drug efficacy, such like sensitive or resistant, which represents a very high degree of drug efficacy or very low degree of drug efficacy. You can also use a quantitative measurement, such like an IC50, which is a very common used measurement to measure the drug efficacy. And the identification of a CDR and the cancer drug response is actually a very crucial problem for the personalized cancer therapy. As a note that the drug efficacy might vary due to the heterogeneity of the cancer patients. So actually this is a very challenging problem. As we know that there are already millions of compounds in the world, but while there is only a very small minority can be considered drugs, the FDA has already only approved about 3,000 to 4,000 drugs so far. So the structure of the compounds is very important to explore. So in this talk, okay, let's first make a very short review on the previous methods for predicting the CDR. So there are actually 2 types of methods. The first type is called network-driven methods. So the core idea is to construct a similarity-based model and design a similarity profile of a known drug to a new drug if the 2 drugs are very structurally similar. So these are very old, and there's already several methods which have been proposed, but there's one very major limitation of this type of methods. And they're typically very low scalable and very low efficient. And the second type of this method towards this problem is the machine learning method. So for the machine learning method, it's kind of very easy to understand. The machine learning models directly take large scale profiles of both drugs and the cancer cell lines as the input. And after they learn the features of both drugs and the cancer cell lines, they use these features to predict the CDR, the cancer drug response. And there are already many models which have been already proposed, and they are based on many different machine learning models, like the linear regression, like the SVM, the random forest, and also the deep neural networks. Okay, so let me talk about some limitations of the current methods. So for the perspective of the drugs, most of the previous methods, they just use the engineered features of drugs, such like the molecular fingerprints. The molecular fingerprint feature is actually a very sparse feature, and some other methods they use the kind of like the smell sequences. They also have some limitations. And for the perspective for the cancer cell lines, most of the previous works they just use the single omic data, such like the genome mutation or the gene expression. And here's our okay. Here's sorry, how to go back. okay, here's our motivation and intuition. We think that each drug can be naturally represented as a graph, and it has a unique topological structures. They have edges and nodes, which correspond to the chemical bonds and atoms. And for the perspective of the cancer cell line, we can currently the multiomics data have been accumulated. So this kind of multiomics data can be used. They can reveal more biological information than the single omic data. So we also want to identify the molecular signatures that can determine the drug response. So this is our basic motivation and the intuition. So okay, let's talk about our methods. And we propose this deep CDR method, which is an end-to-end framework for predicting the counter drug response. And you can see this is a whole picture of our model in the deep CDR. It takes actually two parts of inputs. From the above row, you can see that it takes a drag as an input, and then the drag is represented as a graph, and then this graph will go through a uniform graph convolutional neural network, which I will talk later. And for the second kind of input is from the multi-omics data. So each type of omics data will go through a neural network, and then the high-level features of a different type of omics data will be calculated together with the drag features, and then they will go through a 1D CNN, and then you can make a prediction based on the high-level features. So this is a very simple idea. You just combine the two types of features from the drag and from the multi-omic data together, and then make a prediction. Okay, so let me first talk a little bit about the graph convolutional neural networks. Our model actually is based on our work, which was proposed in ICLR in 2017. Actually, in the propagation rule is actually quite simple. You can say it contains many nodes in a whole graph, and you use a very simple rule to make the layer propagation. You can say that D tutor actually denotes the degree matrix, and the A tutor denotes the adjacent matrix, and X is the facial matrix, and the theta is the trainable parameter. This actually is a representation. So you use a very simple rule to represent this graph, to propagate in this graph. But for our problem, each drag has its own graph. So we may need to do some modification of the original work. So we proposed our uniform graph neural network. So each drag can be also represented with its feature matrix and the adjacent matrix. So what is the feature matrix? Also, actually, each row of the feature matrix denotes our feature vector of our atom in this drag. So it actually are a 75-dimensional feature vector. It includes the chemical and the topological properties, such like the atom type and degree and hybridizations. And as I said before, the original GCN actually is aimed for proposing graph data within a whole graph. But for our work, it contains multiple graph. Each drag has its own size and structure. So we actually derive some mathematical equations and extend the original framework, which it can be listed below. It looks a little bit complicated, but the basic idea is to introduce some complementary graph. So we also try different strategies of initialization is a complementary graph. So with complementary graph, we combine the original graph and the complementary graph together, and then we can make a uniform graph convolutional neural network propagation. So maybe the equation looks a little bit complicated, but this can be largely simplified with some basic assumptions. So the basic idea is that each drag with variable sizes and structures, it will go through this uniform GCN, and then it will be embedded into a fixed dimensional vector. The dimension is 100. Okay, so for the data set, all the data set were downloaded from a public database, such like the PubChem database and the CCLE database, and also the GDSC database. The total training size the total data set contains more than 200 drags and more than 500 cancer cell lines. So each cell line corresponds to a TCGA cancer cell type. So okay, so let me talk about I will accelerate. let me talk about some results. So first, we compared our method with some baseline methods, and for the predicting the cancer drug response, it can outperform other baseline methods by a quite large margin because we use more information. We use multiomics data. Okay, so and also design some blind test, which means that each cell line or each drug only appears in the training set or the test set. So which is a more challenging task. So our model also outperforms the best baseline models. Okay, so and we also the previous two slides using the regression experiment, and in this experiment the IC50 values were actually binarized according to some thresholds. So we can also do a classification experiment to determine whether this drug is sensitive or resistant. So we compared to some baseline method. Our method also achieved the state-of-the-art performance and across different cell types and across different cancer types. Okay. So, and last, we also did some ablation studies, and this is very important because we use multiomics data. It contains genomic data and transcriptomic data and the epigenomic data. We made ablation studies, and the conclusion is that it's very interesting. We find that the epigenomic data and the DNA methylation data actually contributing most, but this type of data was ignored by most previous methods studies. We also evaluated the contribution of the new graph neural network by replacing the adjustment matrix with some identity matrix, and this also shows some power of a graph neural network. Okay, and the last, like to also we did some model interpretation. We propose a very simple strategy to calculate the importance score of each genes in the gene expression data. So we can find some interesting results. So for some predicted results, we find that some top cancer associated genes, they are corresponding. The drugs are already verified to be these genes inhibitors. So we show some biological findings in our work by our work. Okay, I'd like to make a very brief conclusion. We propose this deep CDR as an end-to-end deep learning framework for predicting the cancer drug response. And the major contributions that we proposed our new architecture based on the previous one and made some extension, and also taking the multiomics data into consideration. And deep CDR has some potential values in guiding the disease-specific drug design. Okay, I'd like to thank Professor Jiang Rui and Dr. Zhou Mu and some members in the SCIM company for their help. Okay, yeah, I think
so,
I think that's my basically my talk. Okay, I will first look at some.
So thank you, Qiao. We have a couple of questions in the panel. So the first one was by Deso, who said, how have you modeled the cell lines? In my experience, if you use a vector which simply tells which cell line you use performs really well, meanwhile, it does not capture any biology for future clinical use.
Doesn't capture any biology if you use a vector. Which simply tells which cell. And the cell line I used in our work is actually from our public database. They performed the experiments and they defined many cell lines, and they did then they use a drug. They add some drug to this cell line and and measure some profiles of the cell line. I think these are quite standard cell line. I didn't use very directly using the cell line data. I think it's from the CCLE database. It's a very common user database. But from other similar studies, they are also using this kind of database. It's a very huge database, contains more than about 100 cell lines. And we did some data filtering, and as I said in my slides, we finally connected more than 200 drugs and more than 500 cell lines in our study.
Okay, thank you. So then we have a question by Pau, who says that methylation changes with age. So whether you make any correction for this?
Okay, it's a very good question. We didn't consider that far. The methylation data we also collected from the CCLE database. The next methylation data, I think, is it's also it's in vivo data measuring cell level data, I think. So we didn't consider any age effect.
Okay.
So yeah, it's a very interesting problem. It's a very interesting question.
And the last one is whether you made any investigations such as changing the features of the track, like adding one group or removing another group from the structure of the track.
Some molecular figures, like remove,
like if you remove one amino group from your drug, will this change the profile, of course, of the molecular profile, and will this change? I mean,
yeah. This is a very good question. This question about how we represent the feature, how we represent the feature of a drug. So actually, we have did some kind of for other representation of drugs, such as XML sequences or molecular fingerprint. If I change the amino sequence a little bit, the result will be, the feature will be totally different. But for so we proposed a graph neural network to represent the feature of a drug. So in our experiment, they seem to be a more robust representation of the drug. Yeah, this is a very good question. Thanks.
Okay, so thank you for your answer. So we need to move on to the next speaker, who is Joanna Ficek from ETH Zurich. So whenever you want, Joanna.
Thank you. Thank you for this introduction. So I would like to talk to you about today about SCIM, Universal Single Cell Matching with Unpaired Feature Sets, which is a framework designed to integrate multi-omic single cell datasets. So what is the motivation for our work? Recent technological advances enabled studying samples such as patient's tissue at cellular resolution. Typically, a sample consists of different cell types such as T cells, B cells, tumor cells. Here, they are represented by different colors. Although it is a heterogeneous cell mix, most cells will be present in several copies. In order to study the sample on various molecular levels, multiple omic technologies can be applied, here represented by shape. They can provide information either about gene expression, protein abundance, or spatial and morphological information. However, molecular profiling with each of these technologies often consumes the sample, and the exactly same set of cells cannot be profiled with yet another technology. This makes the integration challenging, since the data after multi-omic profiling, although they come from the same sample, they become a set of disjoint datasets, and they lack both cellular and feature correspondence. And the integration is challenging since most of the available multi-omic integration methods rely on such correspondence. However, recovering the lost cell-to-cell correspondence is possible, for instance, with SCIM, which stands for Single Cell Integration via Matching. SCIM assumes an underlying latent structure that is shared across technologies. This assumption is valid in all kinds of experiments where a sample is uniformly distributed across omics technologies for analysis. SCIM integrates single-cell data by firstly constructing a technology invariant latent space using deep learning, and secondly, matching the cells across technologies using the obtained embeddings. Let's see each step in more detail. In the first step, a latent space is constructed using a set of autoencoders, one per technology, and the technology-specific latent representations are aligned with each other. The encoder and decoder networks are technology-specific, and their architectures are tailored to the respective data types. The encoder maps input data into the low-dimensional latent space, and the decoder reconstructs the embedding back to the input space. The loss is a weighted sum of two components. The negative log likelihood measures the reconstruction error and ensures faithful representation of each technology. The second component is an adversarial term to align the latent representations across technologies. For this, we use a single discriminator parameterized by phi that classifies the latent embeddings into their original technology, here indicated by the shape. By fooling the discriminator, the latent space becomes aligned. This adversarial autoencoder framework constructs a technology invariant latent space, and importantly, it does so without any correspondence in the data space. When trained successfully, SCIM produces embeddings that are well mixed and reflect the underlying structure of the original sample. Finally, SCIM scales to large datasets and feature spaces by gradient-based optimization schemes. SCIM is an adversarial model, and training such models can be difficult. To help train SCIM, we address two major challenges to optimization: orientation of the latent space and model selection. So what do we mean by orientation of the latent space? Consider, for example, that by using a VAE on one technology, we obtained the following latent substructure defined by the colors. Now, we'd like to align the representation of another technology on top, here the hexagons. Since the model is a priori not aware of the labels, the hexagons are in gray. Now the distributions seem to be well aligned across technologies, the discriminator is fooled, and furthermore, the decoder faithfully reconstructs the inputs. However, it turns out that the encoder mapped red cells to the blue space, blue cells to the red space, and the decoder reversed this mapping. This problem arises naturally due to symmetries in the latent space. To overcome this problem, we use semi-supervision to help SCIM align the latent space by providing the discriminator with categorical labels, such as cell type labels. In most applications, only a few labels are enough to successfully orient the technologies. These points act as anchors, defining and coordinating the space around them. The second challenge we address is model selection under adversarial training. This is in general not straightforward, and often qualitative inspection is used to measure the performance of an adversarial model. In SCIM, we use universal divergence estimator that measures the alignment of embeddings between a pair of technologies, here dark and light blue. The universal divergence estimator approximates the symmetric version of the KL divergence between two distributions using only samples from those distributions. In our case, the divergence is estimated using only the latent space samples. It can be used together with the reconstruction error to help model selection by identifying successful runs at scale. Now, in this well-integrated, using the best model and correctly oriented latent representation, analog cells profiled with different technologies lie in close proximity. Therefore, in the second step, these embeddings, marked with Z on the figure, are used by bipartite matching to find cell analogs across technologies. The data from two technologies, here circles and hexagons, can be represented as a bipartite graph where each cell is a node. To increase the computational performance and scalability to large datasets, the search space is first reduced to a set of potential analogs by k-nearest neighbor search across technologies, as indicated by the dashed lines. The Euclidean distance between embeddings is used as the cost on the edges between each pair of cells. To account for the possibility that the cell distribution may not be exactly the same, statistically speaking, across technologies, the KNN graph is extended with a null node. This node is connected to all cells in the root technology and is given a large cost and infinite capacity, allowing for, but at the same time penalizing, any unmatched cells. Overall, it captures the cells for which a correspondence could not be reliably recovered and prevents mismatches. We then phrase the problem as min cost max flow, with the objective of identifying maximal number of cell analogs at a minimal cost. This gives a set of matches represented by the thick lines in the figure. To investigate the performance of our framework, we use simulated as well as real-world data. Firstly, we test SCIM's ability to align substructure in the data across multiple technologies. We use single-cell datasets simulated using the PROST tool with a shared underlying temporal branching process as depicted on the graphic. The branch, which can correspond, for example, to a cell type, is marked by color code and a letter, whereas the fine-grained continuous pseudotime information, which represents progression through some process, is indicated by the color gradient at the bottom. Such a tree-like hierarchical structure is commonly found in single-cell datasets. We simulated 3 datasets with 64,000 cells and 250 features each. The parameters of each dataset's negative binomial distribution were generated using different seeds, So this means that the three data sets have the same underlying structure, but their feature sets follow different parametrizations and cannot be directly compared. Such incomparable feature sets better mimic real-world multi-omic datasets. To integrate the data, we select one dataset as a source technology and initialize the latent space by training a VAE. The resulting embeddings are visualized here using a t-SNE projection with the color code corresponding to the discrete branch label in the upper row and the continuous pseudotime information in the bottom row. Then we run SCIM to embed and align all technologies in the shared latent space, supervising with the branch label. The shared latent space is visualized here by t-SNE, computed on all the cells, and each column shows only cells from one technology. Embeddings of data from different technologies occupy the same region, which suggests that the latent space integration scales well to multiple technologies. Not only data clouds corresponding to different branches in the upper row are aligned between technologies due to supervision, but also the fine-grained pseudotime information in the bottom row is consistent across technology embeddings. In the second step, cells from each pair of technologies are matched using the bipartite matching scheme operating on disjoint embeddings. The density plot shows alignment of the pseudotime information across matched cells between two technologies, source and target. Correctly matched cells are colored with their branch color, and pairs with conflicting branch labels are shown in gray. It can be seen that most branch mismatches occur at the branching points in the tree. Here, the labels are ambiguous since the underlying temporal process is continuous. Overall, SCIM accurately matches cells with the branch label accuracy of 86% and aligns their pseudotime as depicted on the figure, giving precise correlation coefficient of 0.86. We also apply SCIM to a couple of heterogeneous real-world datasets. One of them is a melanoma sample profiled with single-cell RNA-seq and site of technologies, resulting in more than 100,000 cells. Semi-supervision, using only 10% of the labels, is used to orient the latent space. Here we show a t-SNE projection of the integrated latent space using SCIM. A random subset of embeddings is shown, and we can clearly see two clusters of B cells in blue and T cells in orange, with the cell matches marked by gray lines. Despite the unbalanced cell distribution, the bipartite matching results in the cell type label accuracy of 90%. In conclusion, we developed a method to restore lost cell correspondences in multi-omic datasets. In the first step, SCIM constructs a shared technology invariant latent space using a set of autoencoders. In the second step, it matches cells across technologies using min-cost max-flow framework. Thanks to the employment of deep learning, SCIM handles lack of feature correspondence and scales to large datasets and multiple technologies. Such computational integration of single-cell datasets allows to take advantage of the complementary information generated by different omic technologies while preserving the cellular resolution. I'd like to thank my collaborators, especially my co-first author Stefan Stark, as well as our supervisors Gunnar Rech and Kyung Van Lehman. With that, I'd like to thank you for your attention. And if you'd like to know more, you're welcome to skim our preprint, accessible via this QR code. And we also have a small demo on GitHub if you'd like to try out SCIM on your own multi-omic single-cell data. So now I'd be happy to take questions, and thank you.
Thank you, Joanna. So we have so far one question by Fabian. He says, thanks for nice presentation, Joanna. How did you add semi-supervision in the last example? I like the CyTOF plus single-cell RNA-seq, but could you use also match RNA protein expression as anchor? How good are you without it?
So as the semi-supervision, we use the cell type labels. So maybe let me go back to the... yeah, so we select randomly 10% of the cells, for which we give the discriminator the cell type label, so B cell or T cell. But we give it only for this 10% of the cells. So that's why it's a semi-supervision. And we basically, yeah, we append it to the data and in the form of like one hot encoded vector.
Okay, so thank you. We just got another question, so thanks for the presentation. Do this tool works on the single cell RNA and epigenomics, and do this works on the data come from the same cell, multi-assay omics?
Yeah, so we haven't tried on the single cell RNA and epigenomics. I could imagine that the tool should work, right? Because it doesn't require like comparable features. So if that could work on the data that come from the same cell, sure, but the target is to find this missing cell correspondence. So if you have already this cell-to-cell correspondence, then you can use the usual integration tools, right? But the goal of our tool was to restore this missing cell-to-cell correspondence. When you give the two aliquots from the same sample to two different omic technologies, and each of these technologies consumes the sample. So then you're missing this cell-to-cell correspondence. So yeah.
Yeah, it's understood. So thanks a lot, Joanna. So we should finish here and move to the next speaker, who is Marleen Nieboer from University Medical Center in Utrecht. So Marleen, whenever you want.
Yes, thank you very much for the introduction. Indeed, I am a PhD student at the UMC Utrecht, and today I want to present to you about our method SVMIL, which uses multiple instance learning to predict the pathogenic effect of TAD boundary disrupting somatic structural variants, or SVs for short. So let me first introduce to you a bit more about these TAD disrupting SVs. If I can switch my slides. Yes, there we go. Yeah, so these are a type of non-coding SVs, which are in general quite poorly understood. So this is really in contrast to the SNVs, where if you have a mutation in either a coding region or also a non-coding region, we already have a lot of tools that can tell us exactly what the effects are of these. But now instead, if you have an SV, such as, for example, deletion, duplication, inversion, or translocation in a non-coding region, we don't quite know what the effect will be on a gene. And so research over the past few years has shown that maybe these non-coding SVs can actually cause cancer by disrupting the boundaries of the so-called topologically associated domains, or TADs for short. So these are regions in the 3D genome in which you have DNA that interacts more frequently with other DNA inside the TAD than with DNA outside of it. And so here in this figure, you see a schematic where TADs are typically depicted by these triangles. And so you see a left TAD in which a gene is regulated by an enhancer, but this enhancer cannot regulate the gene that is present in the right TAD. And this is due to the presence of a TAD boundary, which is really important in maintaining proper gene regulation. But an SV, like a deletion, can now, for example, remove this TAD boundary. And what you would see is that the TADs can fold open, and now the gene in the right TAD may gain this enhancer and therefore be upregulated. And similarly for inversions, if an inversion spans the TAD boundary, it can flip the DNA around. And what you would now see is that the gene in the left TAD maybe loses its enhancer and be downregulated, whereas the one in the right TAD can now gain an enhancer and be upregulated. And so the big question that we wanted to answer is, do these non-coding SVs that disrupt TAD boundaries now really play a role in cancer? And so for this, we explored a high-quality data set of the Hardwig Medical Foundation, for which we used their breast cancer patients. So we had 162. And for each of these patients, we had both high-depth whole genome sequencing and expression data available. And so what we looked at, so here at the bottom right, you see we looked at the TADs that were disrupted by SVs. And as a negative control, we used each of the TADs that are not disrupted by the SVs, that are immediately adjacent to the disrupted TADs. And so on the y-axis, you see that we computed a Z-score for every gene in these TADs in the patients in which the TAD is disrupted by an SV, compared to all the patients in which there is no disruption by an SV. And we specifically focused on genes for which the SV could cause a gain or loss of a super enhancer. And what you then see is that in the disrupted TADs, you really see a large change in gene expression that you do not see in the non-disrupted TADs. So it seems that these SVs really do something. But what we were really wondering about, because what is quite striking, is that not all genes seem to be equally affected. And we wanted to learn why. So why are these cases different? And machine learning would of course be ideal for this, but it's also quite difficult to properly represent these data in a typical feature matrix. So if we look at this example here, say we now want to know if the SV is pathogenic or not, so we use the SVs as our objects in a feature matrix, then we still need to define what our features are going to be. And so if we look at the example, say we have an SV that maybe now affects 2 genes, and each of these genes gain an enhancer, and maybe one of these genes also gains an EQTL or even loses a promoter, then it's quite difficult to make a lossless representation of all these changes that happen in the genome in a standard feature matrix. And so this is where multiple instance learning comes to the rescue. So this is a bag-based classification technique. So you have positive bags and you have negative bags. And in our case, a bag is an SV gene pair. So this is every SV and a gene that it potentially disrupts. And so you then fill these bags with things called instances, which in our case are regulatory elements such as enhancers or EQTLs that the SV disrupts. And so each of these regulatory elements or instances then has its own feature vector, as you would see on the right. And these features, they can be things such as, is this enhancer, for example, now gained or is it lost for a gene as a result of the SV? And we also included a lot of regulatory information, such as histone marks. So if this instance is an enhancer, do you, for example, also see that it overlaps in the genome with a histone mark that indicates that it is an active enhancer or an inactive enhancer? And so what multiple instance learning then tries to do with all this information is it tries to look for a region where all the positive bags overlap, but do not overlap with the negative bags. And so this region is also called a concept. And now if we look at this particular example, maybe we would see that all the pathogenic SVs, so the positive bags, seem to cause a gain of active enhancers, for example. And so there's multiple techniques that you can use to look for this concept. But the one that we used in particular is called MILES. And it tries to find it by computing a similarity between bags. So here we see a similarity matrix that is used. So on the rows, you have the bags, so the SV gene pairs, and in the columns, you have the instances, so the regulatory elements. And now you can compute this similarity by, for example, taking the average of all instances in a bag, and then computing an absolute distance from this average instance to all of the other instances. And basically what you would expect to see is that your positive bags are now really similar to the positive instances, but they are very dissimilar to the negative instances. And if you would then represent this in a typical feature space, what you expect to see is that positive bags are very similarly clustered together, but are very far apart from the negative bags. And this is great because it means that we can now train a standard classifier in this space, such as, for example, using a random forest as we did. So this solves our representation problem, but the thing that we haven't defined yet is what are we going to use as labels, because we don't know the ground truth of which SVs are pathogenic or not. And so this is where the expression data from a data set really comes in handy, because we can use that to determine if a gene from an SV gene pair, so from a bag, is now significantly differentially expressed in the patient that has the SV compared to all the patients that do not. And so that is what we used as our labels. Okay, but then we are curious, so how well does it do? And we aim to test that by mimicking a setting in which a patient would come into the clinic and we want to know if they have pathogenic SVs or not. So we use the leave one patient out cross-validation. So here in the figure, you see the ROC curves for our multiple instance learning model for each of the SV types individually. And you see the results for the breast cancer dataset. And what we see is that it actually performs quite well. And we did the same thing in the ovarian cancer dataset from Peacock, and then we see that it performs similarly well. So it seems that our method is also really applicable across different cancer types. And now, so another thing that we wanted to know, so how well does SV male now compare to state-of-the-art methods to predict SV pathogenicity? So here at the top in blue, you see highlighted SV male, so the different shapes are for the different SV types. And then at the top right in red, you see a random forest, but then without using multiple instance learning. At the bottom in yellow, you see VEP, and in gray, you see SV score. And what we see here is that on the x-axis, the false positive rate is actually quite low in many of the existing methods. But if you look at the y-axis, you see that the true positive rate is also very low for existing methods. And this is actually where SVMail now is really different. And what this shows to us is that the existing state-of-the-art methods are currently just not looking at non-coding SVs very well yet, while we have now seen that this is actually a very interesting thing to do. And so the final thing that I want to showcase to you today is what we can now do with these with these multiple instance learning models. And that is to actually look at which features are now overrepresented in these pathogenic SVs. So here we are looking at gains of regulatory elements. And on the axis of the circle, you see all the features that we used in our model, so no need to read them all. But the blobs that you see inside the circle for a different color are the different SV types. And basically what we now see here is that when a blob is more towards the outer edge of the circle, it happens more often in pathogenic SVs than expected by random chance, whereas when it's more towards the inside of the circle, it happens less often than by random chance. And this showed us that in our breast cancer data set, these pathogenic SVs really often cause gains of enhancers that have histone marks that show that these are active and that they are also present in open chromatin regions. So I think it's really cool that we can now use these models to start looking at which mechanisms these pathogenic SVs now use to cause cancer. All right, so then to wrap it up, we've seen that these SVs that disrupt tab boundaries can now cause gains and losses of regulatory elements that may change the gene expression inside TADs. And our method SVMail can use this information to identify which SVs are pathogenic. And as we just saw, we can use these models to now get a feel of what the characteristics are of these gains and losses that are most often caused by pathogenic SVs. And then finally, what I did not have time to show you today is that if you look at the genes that are frequently affected by these pathogenic SVs, and you specifically look at the ones that are known to be cancer drivers, what we very often see is that other patients instead have SNVs or CNVs disrupting the same gene. And therefore, it also seems that these non-coding SVs really appear to be alternative pathways to maybe change gene expression in cancer. And so I think altogether, we've seen that these non-coding SVs that disrupt TADs do seem to be playing an important role in cancer. And I think it will be really interesting and also important to start investigating them more in the future in cancer datasets. So with that, I'd like to thank you for your attention, and I'll be happy to answer any questions.
So thanks a lot, Marleen. So far, we have no question on the panel. So may I ask you one question? So
sorry, I cannot hear you very well.
You cannot hear me? Sorry. Sorry, can you hear me now?
Yes, that's good.
Yes, sorry. So we have no question in the panel, so I'm asking you one question. It's about these SVs that relate to this tertiary structure of chromatin. So probably this is not the same in all types, in all cell types. So do you think then the effect of these mutations will be, context dependent? And does your method maybe allow us to predict what context will this mutation be pathogenic or not, like in different tissues and so on?
Yeah, so the features that we use, like for example, the histone marks, are also in the breast cancer case specific for breast tissue. So we really try to take into account if it occurs in the same tissue, for example. So if you would run the method on a different cancer type, then you again use features from a different tissue. So then you can also get insight into what the difference are between the different cancer types with that. Yes.
Okay, thanks. And we have now one question in the panel from Pau Erola. What does mean that SV happen less than expected by random?
So
the biological meaning, he clarifies then, yeah.
Yeah, so I think this probably refers to the last figure that I showed when I said pathogenic SVs features happen less often than random. So basically what we did there is we looked at the, so we have this similarity matrix that we use, which is backed by instances. So basically the instances are there, your features. And so if you feed that into a random forest feature importance, then you basically rank the instances by how often they occur. And so you can look at the top 100 instances and say, okay, so if these very often are, for example, active enhancers, then that happens in the pathogenic SVs. So by random chance, what I then mean is I randomly sample 100 instances from positive and negatives, and I see if in the ones that are really pathogenic, if it's different than in the random instances.
Okay, we have a couple more. So you need to compare two high-C samples. What is your resolution?
So for the high-C data, so we only use this in our features to see if these regions are most often affected by these gains and losses. And I think there we used around 5 kb for that. Yeah.
Okay, so thanks a lot. So we have more questions, but we don't have more time. You can answer these questions, in the panel if you wish. So thank you again, Marleen, and we should move to the next speaker, who is Luca Nanni from the Politecnico di Milano.
Can you hear me well?
Yes.
Okay. Hello to everyone. I'm Luca Nanni from Politecnico di Milano, and today I'm going to present you our work on exploring the chromatin conformation and gene co-expression through graph embedding. So, okay. In our work, we focus on the relationship between the chromatin conformation and gene co-expression. Co-expression of genes can be usually modeled as a network, and an edge of this network represents the fact that two genes have a similar transcription profile. And co-expression can mean a functional relation, or they belong into a shared regulatory program, or they belong into the same pathway. On the other hand, chromatin conformation has shown that chromatin conformation studies have shown that the genome falls in a hierarchical manner, and thanks to the IC, we can now probe unique network for the entire genome, showing all the pairwise interactions between regions. And so in our work, we focus on the question, what the relationship between gene co-expression network and chromatin interaction network. And we modeled this issue as a prediction task, where we try to predict if two genes are co-expressed given their topological representation in the chromatin interaction network. It must be noticed that the relationship between these two systems is still not well understood in the biological literature. And so here we try to shed some light by using some clever encoding of the topology of the network. So this is the overview of our pipeline, and it is composed of 3 big steps. In the first one, we generate a gene chromatin network by using IC. So each edge of this network represents the fact that the two genes are in physical proximity in the 3D space. After that, we generate node embeddings on this network of genes, and we studied 2 different methodologies to do so. The first one based on SVD, and the second one based on random walks. And in the third step, we used the gene embeddings which we generated from IC to predict if the 2 genes we are studying are co-expressed or not in an independent co-expression network. And for that, we used a simple random forest binary classifier. So the first step, and probably the most important one of our pipeline, is the generation of the gene chromatin network. And this is done by binning the IC at 40 kb resolution. And then for each gene, we extract its transcription start site, and we map it to the corresponding IC bin. This generates a genes times gene matrix, where for each gene for each gene pair we have the number of contacts between the bins I and J in the matrix. Finally, we filter out low interactions by taking only the top 80th percentiles of the interactions across all chromosomes. It must be noticed that we use iterative correction normalization on IC before doing all of this. And this has the nice property of preserving the bias towards the diagonal of the IC. And this correlates pretty well with the known fact that genes nearby in the linear space tend to be co-expressed. So we take this into account in this way in our work. So the second step of our pipeline concerns the generation of node embeddings, so gene embeddings in our case. And we studied the singular value decomposition method on one case and a node embedding based on random walks on the other one. So in the first case, we basically performed the singular value decomposition of the adjacency matrix of the network, and then we sum the 2 factor matrices that we obtain from that. In the second case, instead, we simulate a series of random walk and which transit on the interaction graph. And then we find the minimum of the loss function, which basically tries to predict the neighboring nodes to the current one. And this is based on a model called skipgram. And this is generally quite a well-known methodology in graph representation learning. To generate the embedding of a pair of genes, we studied 3 different methodologies. We take the average of them, we take the Hadamard product, which is simply the element-wise multiplication of them, or we take the weighted absolute difference of them. And in our study, we found out that the Hadamard product was the best performing one. So it's the one that I'm going to show in the results section. Finally, given the pair of genes representations, we trained a random forest classifier to predict if that pair of genes was co-expressed or not in a particular co-expression network. And we trained our model on a set of independent RNA-seq data coming both from TCGA and GTEC, and the generation of co-expression was simply done by taking the Pearson correlation coefficient of all the pair of genes and then thresholding these values, taking all of them above the 90th percentile. Every model was evaluated in 5-fold cross-validation, and also we used a holdout dataset at the end, which I'm not going to show, but it's going to show it has similar performances as the 5-fold cross-validation results. We evaluated our pipeline on 12 different matched IC and NSX datasets, and they vary from tissues, cancer types, and cell lines, and also the different resolution in IC and also a different sample set size in RNA-seq. The evaluation was done in 3 different training setups. The first one, we used 1 network for each chromosome, so we studied 22 separated networks. In the second case, we built a single network for all chromosomes, but we excluded interchromosomal interactions. So we have 1 big network with 22 connected components. And finally, we also included interchromosomal interactions. So we have 1 big network with everything connected together. We compared our model against 2 baselines, a simple distance-based predictor, which is simply the linear distance between the 2 genes, so to study the initial property that I discussed, and also a set of handcrafted topological measures on the interaction graph, which are the shortest path between the pair of genes, the Jacquard index between their connected genes, and the average and absolute difference of degree betweenness and clustering coefficient between the genes. So in the first evaluation setting, we can see that both the node-to-vec and the singular value decomposition embedding outperform, of course, both distant and topological measure embeddings. And interestingly, the distance-based predictor can only do slightly better than the random predictor of 50%, showing that in any case, we need more complex and more comprehensive embeddings of topology to discover this relationship between chromatin conformation and co-expression. In the second setting, instead, we still have node-to-vec as the best model, but the singular value decomposition method fails to encode properly the topology. This can be due to the fact that this matrix is a big one and with a lot of zeros in the interchromosomal contexts, but still the embedding strategy, one of the embedding strategies outperforms the other 2 baseline in all settings. Finally, we merged everything together and we studied also interchromosomal interactions, but this could have been done only on 4 datasets having enough interchromosomal reads. This is a way harder computational task than all the others, since the network is very big and hard to train on, but still the node-to-vec and the singular value decomposition method outperform the topological measures. So in our work, we have basically demonstrated that the singular value decomposition and random walk-based embeddings can encode the meaningful topological gene information, and that topological embeddings can in part predict gene co-expression. And in general, the node-to-vec random walk approaches outperform the matrix factorization ones. Of course, a better prediction can be achieved by integrating additional information like transcription factor binding and histone modification. And the proposed methodology can be used in the future as an additional step in the creation of co-expression networks. And more interestingly, the gene embeddings that we find can be used to cluster genes together and find unknown regulatory modules, maybe by integrating also expression data in the mix. And with this, I conclude my presentation, and I thank everybody, and I wait for your questions.
So thank you so much, Luca. There were some problems with the sound, but we could understand you with some background noise. So we have some questions. So the first one is by Davide Bau. So thanks for the nice talk, Luca. When filtering the high-C matrix using the top 80th percentile, do you lose any gene in the gene interaction matrix? Will this reduce the power of your method?
Thanks for the question. Well, we try to find a good threshold for like getting an interaction matrix which was comparable to the co-expression networks. In this way, we could have a good training and a comparable and less usable result. So yes, your question is fair, but we didn't evaluate more than 80 percentile and 90 percentile. So probably yes, we are losing some information, but this is was not evaluated in our work.
Okay, thanks, Luca. There's another question by Peter Bram. So related to the previous talk, will it be interesting to model differential high-C contacts between cancer and normal cells tissue and predict differential co-expression between cancer and normal tissue?
That's a really, really interesting question. And actually, we were already thinking about something like that in the past. We didn't continue on that thread, but it's still in our mind to do this kind of differential analysis. So yes, it's a super interesting question.
Okay, thank you. So let's see if we have other questions in the panel. I don't see any new question. Well, yes, another one. So thanks, Luca, for the talk. Do you think that your method will be applicable for the networks of microbial interaction? That's interesting.
Okay, that's this is a hard question to answer. I actually don't know. So surely we can try to work on that. Currently, I don't have an answer for this. Sorry.
Yeah, I guess you will need the same type of input data.
Yeah, exactly.
And then if you have it, maybe you could try to see, but that will be interesting to see. Okay, so I think that's it. We have 1 minute left if someone wants to have another question. If not, I think we can finish here. I would like to thank all the 4 speakers for the excellent talks and for keeping up with the time, and for the audience. I see we have 170 participants in the webinar, so that's really a success. So thank you all, and I wish you enjoy the rest of ECCB. Thank you.